So we will now move on to our next speaker, who is Leo Swadling. So I think Leo is already in the room, so great to have you with us, Leo. He is a research fellow within the Division of Infection and Immunity at UCL, University College London in the UK. His research focuses on identifying T-cell correlates of protection by applying emerging technologies and bioinformatics to the characterization of T-cells at the extremes of controlled or uncontrolled viral infection and post-vaccination. He is funded by the Medical Research Foundation to probe the link between specificity and function of liver-resident memory T-cells in resolved HBV infection, and a Rose Tree Trust Pears Foundation Advancement Fellowship to continue his work investigating T-cell correlates of protection in abortive versus seropositive SARS-CoV-2 infection. So Leo, it's great to have you with us. The title of Leo's talk is Pre-existing polymerase-specific T-cells expand in abortive seronegative SARS-CoV-2 infection. So thank you very much for speaking to us in advance, and we look forward to hearing from you.
Brilliant, thanks a lot. So does that look okay?
At the moment, this is just your preview screen rather than the other screen.
Okay. Can you see the slides now?
It's still the preview, unfortunately.
Okay, let me try sharing again.
Yep, that's perfect.
Okay, brilliant.
Thank you.
Okay, thanks very much for the invite. So today I'm going to talk mainly about the work that we published in Nature in November of last year, and some of the other studies that are related to this work. This is work that I've led as part of Marla Maney's team at UCL, and we're really fortunate to have been able to work on COVID since the beginning of the pandemic as part of the UK Coronavirus Immunology Consortium, and then also as part of the COVID Consortium, which supplied many of the samples that I'm going to talk about today. And so I will introduce that in more detail later. But first, I wanted to start with an observation which led us to do this work, which is that, okay, let's see if I can move the slides. Yeah, so that despite the highly infectious nature of SARS-CoV-2, some exposed individuals appear to resist infection. And so how we determine if someone's infected, so we use PCR to look for the genetic material of the virus at the time that someone's infected. And then we also use antibody tests to look for a historic infection which has induced this immune response. What we mean by exposed individuals are healthcare workers, close contacts of cases, and household contacts of cases. And we've known since the beginning of the pandemic that some of these individuals appear to resist infection, remaining negative by these tests, despite having the potential exposure to the virus. And the question we really wanted to try and address is, are these individuals that remain negative by these tests truly unexposed, so they manage to avoid the virus, or do a subset of these show signs of very early control of the virus, potentially aborting the infection before it can become detectable and induce an antibody response? And so the reason we could try to address this is because of some of the unique samples we had access to as part of the COVID Consortium. And so the COVID Consortium was a prospective healthcare worker study used to intensively monitor healthcare workers during the first wave of the infections in the UK. And so all of the work I'm going to talk about today is after exposure and infection with the ancestral Victoria strain in March 2020 onwards in the UK. And so this was a crowd-funded study which was rapidly set up, which allowed us to follow these individuals from before and during and after exposure. And we were able to intensively monitor these individuals with these two tests, both PCR as well as 5 different serology assays. And so whereas some studies use a single assay or a single time point to define seronegativity, we could use assays looking for both spike, nuclear protein antibodies, as well as neutralization of pseudovirus and live virus. And a lot of that work was done thanks to Anya McKnight's lab at QMUL. And so using this repeated testing of these healthcare workers, we were able to identify infections in about 22% of the cohort. So this really reflects that these were highly exposed individuals. And we published a paper in December 2020 looking at the immune memory generated by infections in these individuals. But today I want to mainly talk about the work we've done looking at these individuals that remain seronegative, despite working in the same hospitals, having potentially the same exposure and similar demographics. To try and asked this question: can we see signs of abortive infection in a subset of these healthcare workers? And so what would we look for in these individuals? And we've had two nice talks now about both CD4 and CD8 T cell responses. And we now know about the importance of T cell responses in the early control of SARS-CoV-2. So early T cell responses have been associated with rapid viral clearance, as well as asymptomatic and mild disease, with dysfunctional or delayed responses being associated with severe disease. We also know that T cell responses can be detected very early on after vaccination, before antibody responses are detected in the blood and potentially associated with early vaccine efficacy. And this is a paper that we published, that was led by Madinor Zadegi's lab at UCL, here showing, looking at T cell responses even preceding PCR positivity, showing that we can detect by TCR repertoire analysis and proliferative CD8 T cell responses in the blood prior to PCR positivity in individuals that do have a detectable infection. So T cells might be important in early control of the virus, but is there any precedence for abortive infections for other viral infections? And so Marla and I previously worked on viral hepatitis, and for hepatitis B and hepatitis C, as well as HIV and Japanese encephalitis, T cell responses have been described in highly exposed but seronegative individuals. So these are mainly cross-sectional studies looking at healthcare workers after needle stick injuries, household contacts, and intravenous drug users. So this is T cell responses without antibody responses. What's quite unique about SARS-CoV-2 is we know when SARS-CoV-2 started to circulate in humans. So we know roughly when the first potential exposure could be for these individuals, unlike some of these other cross-sectional studies where you don't know the full infection history of the individuals. So this means we can start to ask the question, is there evidence of abortive infection with SARS-CoV-2? And if so, is this outcome associated with particular T cell specificities? Is it favored by pre-existing cross-reactive T cells? So our hypothesis would be that an early T cell response gives the immune system this head start in the race against time with the virus. So if you think about an acute resolving infection where you're exposed to the virus and you have exponential growth, you have a delay as you prime your antibody and T cell response, and this coordinated adaptive immune response clears the virus and leads to immune memory. What might be happening in abortive infections is a very early and strong T cell response can blunt viral replication, stopping it from becoming detectable and from inducing an antibody response. So in this work, we were able to show two independent pieces of evidence to suggest abortive infections occur in a subset of seronegative healthcare workers. The first of which was T cell immunity, and like many other groups around the world, we've used the highly sensitive interferon gamma ELISpot assay to look for antigen-specific T cell responses. And as has been discussed since the beginning of the pandemic, you can detect cross-reactive pre-existing T cell responses in pre-pandemic samples. So these are samples taken before SARS-CoV-2 circulated in humans, but you can detect T cells which can cross-recognize SARS-CoV-2 peptides in vitro. So here we're looking, each column is a different individual, and the columns, the height is the magnitude of the T cell response, and it's colored by which part of the virus these T cells are recognizing. So using highly sensitive assays, we can detect pre-existing responses. But what we were really interested to see is that in our seronegative cohort, we could detect high magnitude and broad T cell responses, higher than you'd expect just from pre-pandemic responses. So this is looking at week 16. So after 16 weeks of analysis after potential exposure in the first wave. And these T cell responses were more in line with what we see in lab confirmed infected. So these PCR positive and seropositive individuals. So if you do a quick comparison head-to-head between the pre-pandemic and the seronegative individuals within our cohorts, you can see that these exposed but seronegative individuals had a broader response targeting several viral proteins. And they also had a magnitude of response which was about 5 times higher on average than what you see in pre-pandemics. And so this suggested that exposure alone, without ever having a detectable infection, has led to an expansion of T cell responses in these individuals. But we really wanted to look for some other indication that these individuals had had a viral infection over the time course that we were studying them in the first wave of infections in the UK. And so again, with Madinor Zadegi's lab, they showed, looking at people who have PCR detectable infection, that IFI27, a single interferon stimulated gene, was the best at discriminating people at the time of PCR positivity. So they compared many transcriptional signatures of multiple genes and single genes and showed that this was the best at discriminating when people are PCR positive. So we wanted to know, is this also raised, expression of this gene also raised in the blood of individuals who appear to have had abortive infections? So we took the extremes of our cohorts, individuals that didn't have T-cell responses after potential exposure, and those that had strong responses at this week 16 time point. And we looked at their IFI27 signal over the first 5 weeks of recruitment into the trial. And so the seronegative individuals which didn't show SARS-CoV-2 specific T cells didn't have any raised IFI27 signal. But what we saw in the individuals which did have strong T cell responses after exposure, they selectively showed increased IFI27 in the blood. And so this was really the most exciting result for me because it validated the fact that we saw these expanded T cell responses and was a second piece of evidence to suggest that they had had SARS-CoV-2 exposure over this time course, despite remaining seronegative. And so if you think about the outcome after exposure to SARS-CoV-2 from asymptomatic through to mild, moderate, and severe disease, we've described abortive infection where you can see this blood biomarker of infection, expanded T cell responses, but without detectable viral replication by PCR or an antibody seroconversion. So this in itself is interesting, but what we really wanted to do is study the T cell responses in these individuals to understand what type of T cell response might be mediating this very early protection and protection from infection and the associated disease. So the second thing we showed is that T cell responses after abortive infections preferentially target the virus's replication machinery, the first viral proteins to be produced within an infected cell. And so whereas there's been lots of attention looking at immunity to the structural regions of the virus, so the spike, envelope, membrane, and nuclear protein, the targets for neutralizing and binding antibodies, and also the main targets of T cell responses in people who have a detectable infection, we were interested in looking at T cells which target the non-structural proteins. For instance, the core replication and transcription complex, the polymerase, its cofactor, and the helicase. That's NSP12, 7, and 13. Thanks to Antonio Bertoletti's lab for showing peptides, we were able to look at T cells targeting all of these regions in our cohorts. And what we saw is that although, like many other labs have described, in lab-confirmed infection, the responses are dominated by structural specific T cells. These are also induced in people who remain seronegative, but really we were seeing a selective expansion of RTC-specific T cells only in these individuals that remain seronegative despite exposure. And this is suggesting that these may be playing a role in protecting these individuals from having this detectable infection. We also went on to look at a separate cohort of individuals who were close contacts and household contacts of cases, where we had true pre-pandemic samples taken in the winter preceding the pandemic, and showed the same preferential expansion of RTC-specific T cells in seronegative individuals and structural responses in seropositive. So the next thing we wanted to ask is, are RTC-specific T cells de novo primed, or are they very early on in the infection, or are they expanded from pre-existing T cell responses? And so TCR cross-reactivity is preferentially a consequence of peptide sequence similarity. And since the beginning of the pandemic, people have discussed the potential for T cells being induced by similar viruses to SARS-CoV-2, such as the human coronaviruses, and how they may cross-recognize SARS-CoV-2. So in collaboration with Francois Ballou, Cedric, and Lucy at UCL, we took all of the publicly available sequences of these 4 endemic human coronaviruses. We aligned them with SARS-CoV-2, and we looked to see which parts of the SARS-CoV-2 proteome are most similar in sequence to these endemic coronaviruses. And what you can see from this heat map is clearly NSP12, the polymerase, and the helicase NSP13 are the areas of high sequence homology with these endemic coronaviruses. We also went on to compare animal coronaviruses and showed that this was also the case, that NSP12 and 13 were the most conserved across all of the coronavirus family. And in particular, they were highly conserved across all SARS-CoV-2 clades and variants of concern. So these regions are highly conserved, but are they immunogenic? So we went to look in our pre-pandemic cohorts, both from London and from Singapore. And of all the proteins that we looked at of SARS-CoV-2, the proteins that were most commonly targeted by pre-existing T cells was NSP12. And then we also, if you look at the seronegative individuals after potential exposure, NSP12 also dominates the responses in those, suggesting that these pre-existing responses are recruited into the response in these abortive infections. We also did showed that the T cells induced in these individuals can cross-recognize HCoV sequence peptides. But really the killer experiment we wanted to do was to show in these healthcare workers on an individual basis that we could see pre-existing responses which expand over this 16 weeks in seronegative individuals. And we were able to do this. We went back to baseline samples and looked at the size of the response to NSP12. And we showed in some individuals we appeared to have de novo responses, with the caveat that the T cell responses may be below the limit of detection on recruitment, but they had expanded responses over these 16 weeks. But we also saw a large number of individuals with pre-existing responses which were expanded despite remaining seronegative. If you take this group as together and you look at their baseline responses, you can see a significant increase in the T cell responses over the study with no change in control peptide responses to flu, EBV, and CMV. And so the final thing we wanted to look at is, are these NSP12 responses enriched in individuals that go on to have abortive infection compared to those that go on to have lab-confirmed infection? And we were able to show this. So although some individuals which have an infection do have pre-existing NSP12 responses, they're much more enriched in those that go on to have abortive infection, suggesting they're protecting these individuals from having this detectable infection and disease. So in summary, going back to our hypothesis, we think these early T cell responses are blunting viral replication and blocking the virus from establishing an infection, becoming PCR positive and generating an antibody response. So in summary, we showed two independent pieces of evidence to suggest abortive infections occur in a subset of seronegative healthcare workers. This is T cell expansions as well as blood biomarkers, IFI27. We show the replication machinery of SARS-CoV-2 is highly conserved across the coronavirus family, and in particular across all SARS-CoV-2 variants, and that T cells targeting the replication machinery of SARS-CoV-2 are enriched before and after exposure in these healthcare workers that appear to resist infection and have abortive infections. So just finally, some of the implications of this. So we've shown an association of these RTC and in particular NSP12 responses with a protection from detectable infection, but their contribution to protection needs to be confirmed in animal models, human challenge models, and vaccine studies, as there could be other explanations for this. So it could be that repeated low-level exposure is inducing this type of T cell response, or that strong innate immunity is mediating protection, and we're just seeing this T cell response as a biomarker of this type of infection. Regardless of that, what we're really excited about is that T cells specific for these early expressed and highly conserved regions do expand in abortive infections and can cross-recognize SARS-CoV-2 and endemic human coronaviruses. So you think these T cells may be particularly effective at early control, may offer pan-coronavirus reactivity, may be long-lived, as we've seen is a characteristic of T cells. And so we think including these in next generation vaccines to supplement spike is a good idea. And so I'd just like to thank all the people who helped set up the COVID Consortium, in particular those at the Barts Healthcare Trust, Antonio for his support, as well as the groups at Imperial and QMUL, which helped with the immunology from the COVID Consortium, all the members of the Maney Lab, and in particular to all the healthcare workers for coming back for repeated sampling. Thanks for your time.
Fantastic. Thank you so much, Leo. That was really very interesting indeed. A story that I wasn't so familiar with. So yeah, thank you for explaining that so clearly. Just like to give you a bit of an update. We're going to have some questions for Leo, so please do type them into the chat or raise your hand, and then we'll be able to go through the questions. I'm glad to say that Professor Liu is back online, so we'll hear the end of his talk in a moment after we've had some questions here for Leo. So please do pop those into the chat or raise your hand. While waiting for those questions, Leo, maybe you can just tell me a little bit about some of the specific epitopes for those RTC responses. Is that something which has sort of been characterized in much detail at this stage? Yeah, so we've done a little bit of mapping. So we've identified some epitopes within all three proteins, NSP 7, 12, and 30, and many other groups have done it in more of a holistic way. Not many people have been able to study abortive infections. So a lot of the mapping's been done in people who have an over infection and PCR positive infection. It's ongoing work to identify all of them and to see if the epitopes that are targeted are exactly the same.